the gated sodium channels open. Movement of sodium ions across the membrane makes the inside of the cell more positive. This reversal of the charge distribution causes the gated sodium channels to close and the gated potassium channels to open. As potassium ions move out of the cell, the original charge difference is re-established across the membrane, closing the gated potassium channels. This sequence of events is called an action potential. The sodium-potassium pump restores the distribution of ions back to their previous levels at resting potential. Let's look at how the action potential is conducted along the axon. As the change in charge difference across the membrane spreads from open sodium channels, other sodium channels farther along the axon begin to open. The original sodium channels close, and adjacent potassium channels open. As potassium ions move out of the cell, the original charge difference across the membrane is restored, and then the potassium channels close. Meanwhile, new sodium channels open, followed by the opening of new potassium channels and the closing of sodium channels. In this manner, the action potential is propagated along the axon of the neuron, eventually reaching another cell. The information carried by this action potential will be processed with other information, permitting this baseball player to make a spectacular catch.
Okay, so the synapse is where the neuron comes in contact, using the term loosely. It's where it's going to pass its message off to either another neuron, to a muscle cell. You can see down here in the corner, this is a chemical synapse between two neurons. Okay, same here. You've got this synapse where the axon terminal is coming in contact with another, whether it's another neuron or, like I said, a muscle cell. And at the synapse is where we're going to release neurotransmitters. Okay, and so we've got a wide variety of neurotransmitters, and we're going to focus mostly on acetylcholine. Oh, it just barely fits. Okay, so we've got a wide variety, like I said, of neurotransmitters. We're going to spend most of our time on one called acetylcholine. Some others that you just maybe should know that they exist are things like GABA, serotonin, endorphins, epinephrine, norepinephrine, dopamine. That's the one that's missing in people that have Parkinson's disease. They're not secreting enough dopamine. And like I said, we're going to focus mostly on acetylcholine because that's the neurotransmitter that's released at the neuromuscular junction, where the neuron comes down and communicates with the muscle cell. So let's take a second and look at kind of the anatomy here of a synapse. So this whole thing is a synapse, and this area here is my presynaptic membrane. Okay, so my presynaptic membrane is going to be on my neuron. Okay, so my presynaptic membrane is there on my neuron, which then leaves my postsynaptic membrane to be on the other side, whether it's on the neuron, whether it's on the muscle cell, okay, but it's on the other side. And in between the two is a gap, okay, a little space, okay, and they're not going to actually physically touch. There's going to be this little gap or space that's called the synaptic cleft. You can see that a little bit better on this one here, okay, so I would have my presynaptic membrane on this side, my postsynaptic here, and that synaptic cleft, that little space that'll separate the two of them. And that's going to be full of fluid, where all of the cells in our body are surrounded by fluid. And before you guys ask about it, I know you heard the giggling and the yelling, that's awesome in the background. That was Ms. Navaja. So when you see her in the hallway, tell her that she's on your video. Okay, so now that we have a basic idea of the parts of the synapse, let's look at what's happening when the action potential gets here. So when the action potential gets here, it's actually going to trigger the opening of calcium ion channels. So all the way down the axon, it's been triggering those sodium channels. And down here in the synaptic terminal, there are calcium channels. And so when it triggers the opening of those calcium channels, calcium's going to head into the synaptic knob. So the calcium goes into the synaptic knob, and that influx of calcium into the synaptic knob actually triggers the release of the neurotransmitter. And the neurotransmitter, whichever neurotransmitter it is going to be released by exocytosis. Okay, y'all remember exocytosis, the fusion of the vesicle over to the outside of the membrane. So the neurotransmitter, that would be all these little green dots in here, will be released out into the synaptic cleft due to that signal of the calcium coming in. And so that neurotransmitter comes across the synaptic cleft here, and it will bind to the sodium channels on the postsynaptic membrane. So now I'm on the postsynaptic membrane, and I've triggered the sodium channels. Okay. And so that neurotransmitter, you can see it's a ligand-gated channel. That neurotransmitter acts as the ligand, binds to the receptor on the sodium-gated channel, which opens that up so that sodium can go into the postsynaptic cell. Okay. And then once that neurotransmitter gets broken down, it will be released from that sodium-gated channel, and it will close that channel back up. Neurotransmitters are relatively quickly degraded in the synaptic cleft. They don't spend a lot of time there because we don't necessarily want to perpetuate and have these really long signals happening. Okay? We want them to get broken. Think about muscle contraction. Okay? If I've got a muscle contraction happening, I don't want it to continue and go over and over and over and over and over again. I want to be able to control that. So basically, as soon as that neurotransmitter is released, enzymes are also released. In the example that we'll be focused on, we'll talk about acetylcholine esterase. Okay, remember, ACE is our enzyme. So acetylcholine esterase is released with that acetylcholine. There, now you can see it a little bit better. So the acetylcholine esterase is released with that acetylcholine, and it starts breaking that acetylcholine almost immediately, so that it doesn't have this perpetual permanent signal. Okay, so the calcium-gated channels were opened up here, triggers the release of my neurotransmitter. My enzymes that break them down start going into the cleft to break them down, and the ones that make it get to bind to the sodium-gated channels on the postsynaptic membrane. And remember, these neurotransmitters they can elicit different responses. Remember, we get different responses, whether depending upon the receptor that's available, the transduction pathway that's happening, the response inside of the cell. So we can have both inhibitory as well as excitatory stimuli happening at the synapse. You can see down here, okay, this neuron has green excitatory synapses as well as red inhibitory synapses. So that could depend, again, like I said, on the neurotransmitter, the type of receptor, the transduction pathway inside of the postsynaptic cell. Okay, but just keeping that in mind, that we can elicit different responses from the same cell. Okay, so don't forget that we're headed outside, so don't wear your fancy high heels and your gorgeous dresses, and if it's cold, okay, make sure that you are prepared to go outside.